Good afternoon, everyone. So, now we will be starting with the live session for the Interactomics course. Hopefully, I am audible. So, in this session, mostly about uh, the different concepts and a little bit about how we can answer the different questions for the assignments which are there for every week. So we will start with the today's with the week one thing. So if you have any other doubt in the start only, you can ask. Otherwise, as we go through, you can let me know. Okay, so let's start. So every week on Saturday from 3 to 5 p.m. Will be this session to solve your doubts of anything uh, regarding the solving the assignments. So for the interactomics course, so as you have, I think you all have gone through the lecture for the week one thing. So you know that the interactomics is one thing which is part of the major field of the proteomics, where you need to understand that the proteomics deals with the identification as well as to of the functional and the regulation of the proteins and their interaction with different uh, proteins, nucleic acids, and other biomolecules, and how they affect for any cellular and physiological processes. So now you can understand that the interactomics helps us to understand all those interactions and their function and their regulation, which helps in the cellular and the which affect all the basic processes of the cells. As you all have, so there are different ways to study the interactions. I think so. You all are aware about it. So, for example, so I just given this introductory slide uh, to just bring you back about the things that what you have gone through the lectures. So just to go through it, so you need to understand that you need to understand in this interactomics thing, you want to understand the interaction of those proteins with different biomolecules, that can be with the protein, with the nucleic acid, with the lipids, with other any biomolecules, or with other small molecules, drugs, etc. So as you have seen that about the tandem affinity purification protein and tandem affinity protein array, so you need to have a bait and the prey protein. So bait is nothing but your query protein, and the prey proteins are your which proteins does it get interacted with. So you know that one way of doing is to bind it to with the promoter regions, so that if both the proteins whichever interacts with those proteins, then it will transcribe, and you can, with the help of the colony selection, you can just choose out those proteins. So these are the different techniques, and also you need to add it with different fluorophores, so that you can understand that with which protein it is getting interacted. So this is just an one method, and I think you have gone through the different methods that are present and that are there, which lets you know that how you can purify as well as you can print the different proteins and different antigens, and you can understand their interaction level, and how much with what chain of proteins does it get interacted. So this is about the just a part of a thing. So firstly, you need to identify your protein of interest, or which protein you want to find out their interactions, and then need to apply through the different processes and methods to try to identify those different proteins. Now, the for to identify your proteins or the protein of interest, or if you want to identify the, so basic applications, or why is the need to study the protein-protein interactions or correlations is that if you, for an example, in any disease condition or any disease state, if a certain proteins where if becomes mutated, or if a protein gets expression gets reduced, then the whole interaction networks, or the with which the proteins is interacting, or with what function it is regulating, will all get disrupted, and that will lead to a major outcome, because you know that the proteins play a major role in your physiological processes and functional output from what it is coded in the genes and the genomes. So once this network gets disrupted, so it will become very much important to know that this particular protein interacts with these proteins interact with uh, which particular proteins or which particular molecules, be it nucleic acid, DNA, or lipids, so that finally you can understand that the disruption of that particular protein can affect how much, what else proteins can affect, so that you can take a measure of it and then try to uh, uh, give the supplements accordingly. So this is the major reason why we need to understand the interactions of the protein and the complex interactions, and also know that the biological processes are not quite static but major dynamic, and the plays of the co-regulation are very much present in any biological processes. So it's becomes very much important to understand each of these things. So now, for basic identification of the proteins, so firstly, if you want to understand and identify the proteins that what is present in your sample, so if you just I'll just go through it once, the different overview of the proteins, identification of proteins. So firstly, it was through different gel based proteomics. You know that firstly the proteins were run on SDS based gel to separate on the basis of the molecular weights. Then you know that the 2D came with O'Farrell, that the proteins can be separated based on both uh, basis of their pi and the molecular weight. So on that basis of that, you can get a certain set of proteins which have a unique pi and the molecular weight, and you can understand. But then if you want to, then the 2D dyes came, and you can understand the quantitatively that which proteins are expressing how much in the disease condition and in the normal condition, that which proteins are expressing high, with the basis on the basis of their expression level you can understand, one could understand. But to get a wholesome picture of all those proteins, all those proteomes in a very robust way, in a very reproducible manner, so then came the um, mass spectrometer. So with the help of the mass spectrometers, one can understand, and uh, one can identify the proteins, as well as identify the expression level of the peptides and the proteins, and can understand that what is different in both the disease condition and the normal condition. So for understanding the interactions between the proteins and the networks, so firstly, what we need to understand that what proteins are we interested in. Also, one thing that the interactions also can be done through the mass spectrometers, where you can pull out certain proteins with an antibody, particular antibody, and then you can also look at that which proteins are present in that particular interaction thing in the mass spectrometers. So mass spectrometers can play the role in both the things. So a basic idea and the knowledge about mass spectrometers, because you know that mass spectrometer based proteomics has helped to evolve this field to a great extent. And after this, what you can do and what you can understand is that once you are done with the mass spectrometry based identification of your particular proteins and those peptides, then you will need to identify their interactions, and through different ways. So for the concept here lies in that that the mass by charge ratio will be unique for each different molecule or different peptides, and the identification of that would lead to the identification of that would lead to of those unique molecules that are present. So the different going before, so you just need to have just a basic idea of the different mass analyzers. So because you need to that it depends on very much that how much uh, yeah, so today's date different mass analyzers are present that help you to analyze all to detect all those ions which of the molecule. So you have your like time of flight mass analyzer, you have ion trap mass analyzers. So these are the different mass analyzers. So in here, when you are having your like the time of flight mass analyzer, so the time to reach from the source to the detector gets detect is recorded, and accordingly you can, so like the same way your based on the molecular weight your proteins uh, what you say that uh, get separated in the gel. So similarly kind of thing. So with the mass 2 based on their charge, it will get separated, and with this unique M by Z, you will be able to detect that what your peptides or what your things are. So after that, with the after those detection of the peptides, you can understand that you can relate to what peptide is according to the sequences. You can then identify your protein sequences. So on the so firstly you can think that with the proteins or the molecules you can just ionize those, and then you can detect those, and then after detecting you can specify your what kind of uh, peptides and proteins are there. So these are some examples. So here like in this quadruple mass analyzer, so it usually runs your selective ejection, where it will just capture your those trap those ions, and then in increasing mass it will increasing by increasing the voltage, it will leave the ions for the analyzing those into mass analyzer. So this is just one example. Another thing which is becomes quite important is to, before the going into the mass analysis, is that you need to ionize those. So this ionizing thing was quite feasible in the chemical things, because there was no such biological materials or available, but controlling the voltage and controlling the heat was quite become very much uh, needed to analyze your particular biological materials, biological small molecules of the proteins. So you need to have those. So after the invention of your MALDI, which is your matrix assisted laser disruption ionization. So here in this matrix, with the help of the laser, you can control your voltage, and as well as it will give you the charged ions, and when it helps you to disruption of the matrix, and then it carries your analyte it into the gas phase. So this, by absorbing the laser light energy, then analyte to vaporize. So this as helps as the proton donor and acceptor, and this helps you to ionize both positive in both your positive and the negative ionization modes. So this way the compounds gets ionized, and it you can like after ionizing those in the of analyzer, you can detect those your ions, and then you can understand that with which molecule it has come. So this is one. Another thing which is generally used is for the electrospray ionization technique. So here in the liquid sample solution, with the help of an uh, electric source, so it gives the charged ions, and then in the presence of the vacuum it gets into the mass spectrometers for the different mass analyzers, and where the ions are analyzed. So in this way, so these are the two major techniques that have been used. So your ionization techniques depends on the what biological materials you are going to analyze, because uh, that can help in means the all the sources and the charged voltages will make your make the molecules either capture it, or it can also degrade it and it won't be detected. So these are the two things, and these are the some examples of the mass analyzers. And the mass spectrometry helps you in identification of those proteins. So it combines both these things with the mass analyzer and the ionization techniques, and you can help in detecting those particular proteins or the molecules. So this is one thing. So I think there is no doubt regarding it. So and understanding. So there are different mass analyzers and the different ionization techniques. So you need to now understand that what kind of ionization techniques will you use for your particular material, because that can hugely impact on your sample and on what you are identifying. And also the different mass analyzers have their different specificity and their sensitivity. So you need to understand that which one to use for your particular uh, set of experiments. And also you can understand with the help of their names that what principle, on the basis of what principles it works, and it helps you to understand. So now another concept is here for the monoisotopic mass. So for example, if you want to understand uh, a particular amino acid or particular peptide, so you can identify their monoisotopic masses. So it is quite important to understand that how this calculated, then, because whatever uh, so in this mass spectrometers there, when you get the two ions, so you will get the identify the monoisotopic mass. Then with that monoisotopic mass you need to calculate the which it belongs corresponds to which amino acids, and then you need to identify the sequence of the peptides. Going to the that deep is beyond the scope for this course. So if you want to understand and learn about more about this, then you can take the other courses which are proteomics or the proteogenomics courses. So you will get the idea of exactly how it is done. So here for the monoisotopic mass, so exact mass of your ion or molecule of the mass of the most abundant isotope of each element. So for example, for H2O, here 2H and H monoisotopic, the abundant isotopes elements weight and oxygen's weight is calculated to give you the particular unit. So for example, if you are asked to calculate for a particular amino acid what is the monoisotopic mass for the particular amino acid, then you need to identify the first formula of the particular amino acid. And after you get the formula of that particular amino acid, so you need to just calculate their masses. So for example, with the carbon, with the hydrogen, with the oxygen, and you will get the monoisotopic mass. You will be able to calculate the monoisotopic mass of that particular amino acid. So because once you get the results in the form of the ions and the masses, then you need to, because you can understand that now why this is needed. So here I told that you will get that in mass spectrometers you calculate and understand the m by z's, right? So m by z, so calculates your mass by charges. So for so whatever you would be detecting would be your m by z. So you charged state will be either plus 1 or plus 2 or plus 3, uh, dependent on your ionization. So it with then you need to trace back with the, so you will ultimately get the m, and when you get the m you need to correlate back that what is your particular element is that. So that is an that is what amino acid is there or what molecule is there, and then you can understand the whatever you are trying to see or identify. Okay, so this is for the monoisotopic mass, calculation of your monoisotopic mass. So this was all about the mass spectrometer, basics of the mass spectrometer. So for the proteomics technologies, so going on through the gel based proteomics to identify the proteins of the whole organism, whole cell, whole tissue, or uh, whole organism, and to identify that which proteins are getting dysregulated at which state. And also it helps you in second. Also it helps you in identification. The proteomics technologies also helps you in the identification so of the different proteins. So then after the interaction, after you know that which proteins have been dysregulated, and also you need to identify that which proteins are exactly interacting with each other, and because of that dysregulation or the mutation or anything that has happened, so which are the proteins that is exactly it is affecting. So that's why you need to understand the interaction of the complex network. So before that, so you can understand that proteins went through the different stages of evolution. So firstly with the gel based, and then the mass spectrometry based. So mass spectrometers helps you in identification of those proteins or those peptides which are getting affected in the diseases. And so here for the basics of the mass spectrometer, so these are the different mass analyzers which are present, like your quadruple mass analyzer, time of flight, and then ion trap, and then your ionization techniques that can be through MALDI or that can be through electrospray ionization, or there are other ways also, but these are the most commonly used. So this concept only came after the MALDI technique, when you could control your voltage for the handle the biological specimens. Otherwise, it was not possible till that time. Then you need to calculate some of this monoisotopic mass, because in the mass spectrometers you will get the m by z charge ratios, and then you need to calculate the masses, and with the mass you need to identify the particular amino acids, to identify those particular peptides and those particular proteins. So now again there is a thing for the, now when we now after we identify, we are now going for the interactions or the interactomics with which the course is. So now for to identify the interactions, so this again started with the DNA microarray. So now there was a subtle difference between the DNA microarray and the protein microarray. So why is that? Because this in so the different things that are used for the DNA. So you know that the DNA is the proteins are quite dynamic, and there's a huge their structure. The proteins won't work on a specific, if their specific structure is lost, it won't interact or it won't show that kind of binding or affinity, whereas DNA is quite flexible in that sense. So they are different. So, and also you know that I think if you have gone through the lecture, you could have known that the protein microarrays, what has been taken from, or has been taken inspiration from your DNA microarray. So what are those techniques that were present? So why these techniques could not be used, or which techniques are now being used? So you will get an idea. So for example, in the DNA, you will get the physical absorption for the charge. So you will just uh, advantage is that you can fast and the direct method. You will just with the basis of the physical absorption. So you will just absorb it by hydrophobic interaction or your charge-charge interaction. The drawbacks is your disruption by change of ionic strength, random orientation, disruption by detergent. Then you have covalent bonding. So covalent bonding, you know that will give you good stability, high binding stress, or used during long term. But you need to have a linker molecule to link. Then it will be irreversible, slow. Okay, crowding effect and all those things will happen. Then your streptavidin biotin interaction, specific streptavidin, so that helps you in improved orientation, and it was very much specific functionality, well controlled, reversible, but it is expensive, slow. So these all the techniques what you will be going through, immobilization techniques or whatever techniques for to identify interactions, so everything has its own merits as well as demerits, but some we have to trade off somewhere, and then we need to choose some of those things where we can uh, go through it and where we can identify. Now with that we have come to the protein microarray. So I was just as I was mentioning, so there you can, the protein microarray, you can screen the antibody, you can perform the enzymatic assess, that where these enzymes work with which particular protein. So if you have got an identify any unknown enzyme and you don't know on which protein it acts or interacts, so you can just have that particular proteins printed on a microarray, and you can just provide that enzyme, and wherever it gets the work done. So for example here ATP, it quenches to ATP, and then you can just detect those molecules, the end reaction, and then you can find out where it worked. Then protein DNA interactions, protein small molecule interactions. So this is quite important, as you know that whenever you apply a drug or anything, so it always acts with particular protein, or it gets bind to a particular protein and try to either stop its role or disrupt its structure, or disrupt it functionally. So as you know that for a particular enzyme, protein enzyme, if you provide a inhibitor which is quite similar to the substrate, then you will try to do a competitive inhibition of that particular protein. And if your small molecules quantity is quite high, then the actual substrate won't be able to bind to your protein, and uh, it will help you in inhibiting or whatever you had have required. And other things are for the protein lipid interactions. Another thing is for the protein protein interactions. So proteins also requires to interact with a particular protein. So whatever interactions will you get. So these are the different things. So you can understand that after the DNA microarray, the protein microarray was evident, and how helpful it was in identifying so many things. So it helps us to give a much more comprehensive view of the cellular and the processes, whichever is there. So, okay, so this is, now we will go through some of this. So if with this review article you would be able to understand in depth more about your uh, protein microarrays, what are the different developments and what are the different applications So this is very much important for you to understand first that. So I think till now you are clear that why protein microarrays are helpful and how protein microarrays are quite helping us to understand the different interactions of the regulatory networks. Now let us look at some of these developments and those technologies, and also if you have any doubt regarding, because in the lecture in the first week itself you have gone through a number of protein arrays and their types and how, what are their advantages and disadvantages. So let us go through it once, and I think then you would be clear about whatever was being taught, and then you can be able to solve any question in that matter. So you can, so I think till now we were clear about the protein microarray technology, so which has the greatest potential on protein function and your potential drug target. So you will help you. So just so now I think you would get the actual interest also, that you would be able to knowing your protein function, as well as you would be able to know your potential drug targets, as well as you would be able to see the effect of the drug on that particular protein, if that drug is actually binding or not, with what kinetics it is binding, or with what how your drug will get away from the is either getting away from the protein, how quickly or how robust the binding is, so that you can get that, you will get the clear picture and the clear view. So, okay, so for example, then your functional protein microarrays for the mapping of your biological pathways, so whatever the pathways are here. Okay, so that takes place involving protein lipid, as I have just mentioned, protein DNS, protein small molecule interactions. So this, and the most important advantage is here, you need a very small sample amount and the potential, and then protein microarrays, it will help you to profile your proteins, drug discovery, drug target identification, and clinical prognosis and diagnosis, right? So one example I would like to just give, which is a recent sentence, I will show you what. Yeah, if you can check that how this is helping, this technology still now is helping and people are using, so you can see that how real life importance, you can see that the real life use for this. So you can see that a protein microarray with serum protein investigation, so in the, for a particular cancer with the basis of the serum, so you can think that in a non-invasive way how you can reveal the autoantibody difference signature in your cancer patients. Okay, so this you can see at that whole proteome you could profile, and then you could get a panel of those particular proteins. Okay, like this. So list of this significant autoantibody signature, so it will help you with going for early detection or whatever we think here, that so it just screening and the profiling of your serum samples of the patients, you can get an idea. Okay, so you can see like this, you will get that in the what is the in the healthy control and what is in the your patients what use. Okay, so what are those different proteins levels? So here you can get a glimpse, right, of those particular proteins which of which the antibodies are generated and their interactions or the interacting network. whatever is their role. So you can get both diagnostic and both prognostic thing. And also so in another thing is also been used. So yeah, so you can check that not only so, and so here different kinds of, with the help of different kinds of tumors with the different autoantibody signatures. So just think that if you can have your one signature panel for each type of tumors or cancers, that you just go on detecting through those patients whoever has a little bit of symptom, and you need to just collect your serum and just go to the clinic. So here you can see there, if you just go through these two papers, you will get to know more about your that application of these technologies. So you can see that here is one particular protein, a panel of proteins for this tumor and another panel of protein for this tumor. So you can have a tumor based panel. So once like you are going for blood test now for different diseases like your blood sugar, lipid profiles, and also one can have a tumor profile like this, where for the serum just you need to print all those proteins in a particular chip, and you, from with that serum, if it is being analyzed through particular chip, so immediately you would be or the clinician would be able to detect that okay, so this patient is having the level of the autoantibody profile is like this. So if it is being prone to a particular disease or not, or if that patient is already diagnosed with, and what is the cases and how in the it is being progressing or not, so you can get an idea easily. So this is one of the major applications of the protein microarrays. So what you can get to it. So now there are different protein microarrays. So one is your functional protein microarrays. So it will help you. So now you just understand that different microarrays. So now you can correlate that these two papers are what kind of microarrays. So for the this does not tell about those two papers. It doesn't tell about the functionality of a particular protein, but it helps you in detection of antibodies. So how they are detected antibodies? They have all the proteins are printed on the slide, and when your serum sample, if it contains antibody for that, for any particular protein, so then it will bind to it. So that is the major thing. So that is your protein detecting microarrays, your lysate microarrays, antibody microarrays, so detection of antigens, detection of antibodies, all those protein detecting microarrays. But if you know too about the functional protein or the protein domains of the protein-protein interactions of the small molecules interactions, or your enzymatic analysis, that like that, which these enzymes happen, which protein or what function does it play. So this you need to have your functional protein microarrays. Whereas if you want to detect the different things, then you need to have your protein detecting microarrays. Okay, so these are the two different. So that thing corresponds to your protein detecting microarrays. So that can be your direct protein arrays, or your sandwich amino acids, or the arrays whatever was there. So that is your detection not your functional protein microarray. So now the functional molecule, if you want to understand the network of the molecular interaction, so now it will depend upon your application or what objective you want to perform. That if you want to perform the network of the understand the network of your molecular interactions, or you want to like to perform a diagnostic test or a diagnostic potential wala thing that help you in understand in help you in diagnosis for a particular disease. So it will depend on that. So here you can see that the network of your molecular interactions, so that defines a particular proteome. So this will help you in understanding the strengthening the functional proteomics part. So here what it helps is that extremely, so you cannot, there are 20,000 proteins for example in humans, so similar different huge number of proteins for particular organism. So you would not be it was not possible to screen one particular protein. So this has been overcome by the help of the functional protein microarrays. Okay, so here you can combine recombinant proteins and chemically slides, and then without affecting their molecular and functional integrity. So one important thing to understand for the protein function is that you you cannot if you want to understand or assess the functional proteins functional part, so then you cannot affect their molecular structure or functional structure. Okay, so these are the different beginnings of how the proteins microarrays were developed and how this started. So then protein chip was evolved with different, so it also got evolved with the time, so to increase its functionality or the robustness, so that the proteins gets in the chip in a very clear way and it does not get break down and gives you the best result. Okay, so the microarray dataset in combination with protein-protein interaction and the transcription factor binding data. So different regulatory models in yeast. So not only in the human but in the model organisms like yeast, Arabidopsis. So everywhere it started, people started using it. So to get the knowledge about the proteins and their functional roles and everything. Okay, so analysis of your. Okay, so now then another thing, important thing is that functional protein domain microarrays, it can also help you to quantify your protein interactions. So if you want to quantify your protein interactions, okay, so by using your frozen level, it will help you in identify the differential equilibrium. Okay, so similar approach to quantify the interactions of several human with different phosphatidylation containing peptides derived from human receptors. So this type of protein mining need better study the binding properties of complete families of protein and the protein layer domains. Okay, so this is one kind of thing, so you can check it out. Then is your protein detecting microarrays. So once your one is your functional protein microarrays, then your protein detecting microarrays. So functional microarray protein array, what will help you to identify high throughput screening and quantification of protein interactions. And whereas but in the detection microarrays are ideal reagents for this type of, but if we use the monoclonal antibodies also specific protein captured reagents. So I am just stating all this so that you can visualize the things and can understand the things that why these are necessary. Okay, so the antibodies, you know that it's are quite specific in nature, and so you can also think whenever you come across any problem or particular disease or anything, what if you want to assess the, for example, you want to assess the quality of your drinking water. So if there are any pathogen present or not, so if you just take those water and with the help of in the array, if you have the pathogenic organisms, uh, protein or antibodies or anything, then you can just detect that okay, so these pathogens unique proteins present in the water, then it is safe or not. So in these applications also this can be used. So this is a wide spread application and widespread use, so to be used in different things. Okay, so these are so specifically antibodies are were generally used, but so and also this going through this you would understand that why in NAPA or selfie system came into existence, or what was their advantage over those classical antibody just arrays. So because so it's quite natural that antibodies are quite specific and quite robust, but the potential associated with was your cheap assembly, okay, through moderate expression use, then the stability solubility, so then it led to you new then then it became quite costly, quite laborious, and quite generation of all those antibodies, and then those placing it on the chips, so this all become quite. So then it changed. So so in this antibodies, your if you remember from the lecture that the protein microarray for the direct protein, it can be captured with the protein is stacked with the frozen, or the sandwich type of thing where after you give the antibody, then the antigen, then another secondary antibody with the fluorescent tag. So the fluorescent tag is linked with the secondary antibody, and that uh, gives you the identification of this. Okay, so then here you can see, as I told you, a serum protein profiling to identify new biomarkers. So you can see, so I showed you two papers, and also there are different papers which have told about height to serum protein profiling through microarray is quite known technique and quite robust technique for specifically for when you want to analyze for the particular biomarkers or for early diagnosis things, so that in a non-invasive way it becomes quite evident and quite useful in nature. Okay, so now, but the availability of suitable antibodies for capture and detection are not quite feasible. Okay, so use a multiple fluorescent which background signal as well as the cross-reactive binding as a member of antibodies increases. So once you go through it, you can understand the different things. So now, so how you will understand the differences in the particular two groups? So you need to identify through the different photophores, and the different photophores will give their different intensity, and you will detect it through the detectors, the different photophores, and with from their intensity level you will be able to identify. Okay, so now the thing is that although antibody microarrays, so this is quite important. So when the antibody microarrays are working well, behaving quite well, so why it was not needed to change it into different other kinds of microarrays, or why it is needed? So, okay, so this is mainly due to your lack of available well ventilated well validated antibodies. Is a combination of different with data generated in this work, data from antibody enzymatic flow cytometry to assemble a network of 10,000 interactions. So just imagine that how much interactions could be with different combinations of the cytokines, so how much they affect each other, their regulation. Okay, between pro and anti-apoptotic signals by different cytokines. So then, and also if you directly print the proteins, you know that the protein expression and the purification, these are also become quite a tedious job. So when you expresses your protein in a cell, for example yeast, or for example any bacteria or the expression vectors, so then that contains the host proteins as well. So whenever you are pressing your particular genes or the clones of interest and then putting it into the bacteria, and then you are expressing a particular protein, then your protein will contain along with your protein of interest different proteins of different that host proteins. Then you need to figure it out and change, uh, means purify from the host proteins. You need to first separate your proteins of interest uh, from the particular host proteins, and then you need to collect those, and then in a good amount without destroying or disrupting structure, and then use it in the uh, microarray. So this is for the protein lysate, for the protein expression and the protein microarrays. So then everyone's focus shifted because all this was becoming quite tedious job. So to going for the protein lysate microarrays. So here was to immobilize cell lysates, because cell lysates are quite not so much difficult to handle, as well as it is not so much tedious to generate antibodies. So you can understand, right? So with for generating antibodies, you need to have a huge cost and labor will be associated. Then use specific monoclonal antibodies to identify the presence of a particular. So as I told you that if you can identify and understand that what proteins are what your interest through mass spectrometers or through the gel-based proteomics or through the mass spectrometers for now, if you could identify that in your cells these are the different markers that is getting responding or that is being changing to certain conditions. For example, you are experimenting in such a way that you have make the cells deprived of nutrition, or you have just cut off one nutrition in the cells in the media. So now your cells are stressed. So what changes or what proteins are markers? So you can design an experiment like this. So one set of cells you are giving the full media, and one set of cells you are not giving any particular media, any particular element, for example any essential amino acid or any essential fatty acid deprived in the media. Now the cells you would take and perform your protein expression analysis through mass spectrometry to identify all the proteins and their expression levels. So for the proteomics or the proteomes, you will get that all the proteins what is present in the cell and what is their expression levels. So for the nutrient medium containing cells have a particular expression of a particular protein, as well as in the deprived one has a particular expression level and a particular thing. So what will happen is that you can get to identify and then quantify. So now after that, so you have identified one particular protein which is of your interest. So now you can monitor it across the different cells or different conditions that you trace the cell lysate, and then you will for that specific monoclonal antibody to find its expression level. Okay, so this is technology was that. So this help you uh, in a large number of cell lysates and a large number of samples. The same approach now has been used for disease diagnosis and the patient stratification. So similarly, with a particular protein, if you monitor a particular protein across the cohorts, across the different grades of tumor patients, so then you can use that to stratify the patients. So patient stratification, as you I think you might have known that the different tumors are stratified with the different grades based on their aggressiveness, grade 1, grade 2, low grade, high grade, etc. etc. So you can understand and identify those things. Okay, so then here people have also used lysate microarrays to study your kinetics of intracellular signaling by tracking 62 phosphorylation sites in simulated Charcot cells. Okay, so novel approaches. So then I told you about the protein immobilization, like the DNA immobilization technique there. So here different techniques are also there. So here one is your of the solid support that has been non-specific assumption or your covalent cross-linking of naturally occurring chemical groups within proteins. Okay, so here also the same thing. The first one is your physical absorption that will absorb or your uh, or a non-specific absorption or using naturally cross-linking of the chemical groups. Okay, so this is the key issue for the fabrication. Now that is use of your recombinant affinity tags. So site-specific immobilization, the most commonly used your affinity tags or biotin, avidin, his tag, natural acetic acid, GST, all these things. Then immobilization of your antibodies to the FC region, you know, of the protein E or protein G coated surface. So if you want to immobilize your antibodies which will have an affinity towards your protein A or protein G coupled surfaces. So if your surface has been coupled with protein A or protein G, then what will happen is that your antibodies will bind to that, will have affinity towards it, and it will be used for your antibody microarrays. Or otherwise you have, if you go for the chemistry and the different chemicals, so here it is seen maltose binding protein chitin, so all this can be used. So protein DNA conjugates have also been recently reported for the DNA directed immobilization of proteins onto complementary DNA microarrays. So you can know that with the complementary DNA microarrays it will be able to immobilize your techniques. So but these are not reversible and not your stable over time. Okay, so these are the some things. So recombinant expression, size specific derivatization, chemoselective immobilization. Okay, then of the proteins in micro are your surface modifications. So if you have modified certain regions of the surfaces and you want to, then you can, like the introduction of amino and the thiol groups respectively for the substrates, so then modified appropriate linkers, and then you can are more reactive towards those groups, and then you can solicitate like properties. So compounds containing your thiol or selenol can be used to modify your substrates. So and then is your using express protein ligation. So for the site specific immobilization. So here what is your site specific means on the basis on the location you would just like to immobilize those. Okay, so this helps in the selective recombinantly produced proteins alpha thioesters with surface strengthening in amino residues. So with it will have an affinity towards your N-amino cysteine residues and it will get bind. Then different other techniques, so you just go through it, you will get to know of everything. And then active site directed capture ligands. So these are all novel techniques based on your proteins and the immobilization techniques on the modification. Now comes on the cell-free expression systems. As I already mentioned that in the proteins it becomes quite difficult and tedious to know and to understand that how the protein expressing and then taking out it becomes quite difficult and challenging. So people have generally used is that on your cell-free expression systems. So here you can see that, so based on bacterial or EPI, it is a very time consuming practice that requires large amount of manpower. Moreover, the presence of your disulfide bonds and PTMs etc. So it will require more specialized expression systems, and so all this led to your cell-free expression systems. So the cell-free what you, so you just take the cell extract, and for carrying out need to carry out your transcription and the translation in vitro, and then it will extract exactly from the your E. coli or rapid reticulocytes or the wheat. So this helps you in providing all those factors needed for the transcription and the translation. Okay, so these are required, and so and so these are the ways through which just using cell lysate you can print and mobilize those proteins, and then finally you can find out the expression level changes, whatever you are checking. So like this protein in situ array, so I think you have in the lecture you have heard of it, of the protein in situ array. So proteins are directly from DNA in solution and immobilized as they are produced from the surface through a recognition tag sequence. So basically in situ PISA and NAPA. Okay, so here you can see the cell extract. So protein in situ array is one of your kind of a cell expression free system. Okay, so just try to remember and understand the concepts of it. So there are with the protein arrays, with the protein chip, functional protein arrays, protein detection protein arrays, then you have your cell free based expression systems. Among these are the protein in situ array or NAPA. So here what are the differences? Is your description. Okay, so here coupled transcription or translation protein capture through a tag, and then you will do the wash, then the DNA encoding protein tag capture region, and then recombinant affinity tag. Similar, and this is for the protein in situ array, which will help you to the protein tag with the capture agent, and once you pass to the proteins through it, you will get to identify all those. And here for the cell extract, here you will get the uh, anti-GST tags, or all those tags helps you in uh, so here protein is captured through a tag. So the major difference between your PISA and NAPA is that one is your protein is captured through a tag, and another is your protein is captured through your antibody. So whenever protein is captured through antibodies, so it will then you can analyze the. So here you can see that the protein tag capture agent, and here protein of interest GST tag with the protein, and these ribosomes are translating your cell extract from the cell extracts uh, DNA to cDNA to protein. So in this approach, what you are getting that all proteins are expressed in parallel in vitro transcription or translation systems, and then it is carried out on the surface. Capture agent bind to the affinity tag and immobilize the proteins. So this is usually typically using his tag proteins and Ni plus 2 and K. So if there are any question or means in the assignment or something, so you need to understand those, that what is happening, like for the tap or the affinity purification of the proteins. So here you, in the slide also I have shown you that what are the different uh, things that is getting attached, or what is your bait protein, what is your prey protein, then which the uh, identify the, what you say that if their proteins are interacting, then whether that you will get the blue colonies or not, whether when will you get the white colonies. And so it blue colonies says about what that your, that the operator has, promoter has functioned well, and what your proteins, two proteins are interacting, and it helps in the expression of that beta galactosidase. So this, like this, you need to understand some of the basic concepts and applications to able to answer in your assignments and your questions, whatever become. So you could be able to understand. So here you can see that it is accomplished by using his tag proteins and Ni plus nickel NTA coated surfaces. So affinity, so in your cell expression system has been tagged with the gene which uh, produces his tag proteins, like the proteins containing in the end a series of histidine. And if your capture agent, if you make your capture agent with the nickel into your capture surfaces, then you can just easily capture all those proteins, and then using those proteins you can you can easily run through it. Okay, so this is for the nickel NTA coated means for the histag proteins. So PISA method, as I told you, that it doesn't capture with the antibodies, but captures with your particular capture agent. So why, how it gets the confidence of capturing it with the capture agent is that it you in the protein itself, when you are expressing, you are giving certain chemicals like the histag, so that you can design your coating like the nickel NTA coating, then you can just capture your histag proteins there. Okay, so once so this what is happening is that once the protein is translated, so once it will get translated, it will definitely get into your thing is that it will get translated with the uh, histidine. So then only you will capture it, and then you can easily find it. Okay, so this is one. Then is your PISA method. So PISA method, what is that? That using a small. So this is basically about the PISA method. So this onto the magnetic bead, PISA was used. This is just a brief about that when it was used and with what thing. Okay, so then finally, then after the PISA, then came the major thing about the NAPA, which is your nucleic acid programmable protein array. So that is also allows your on-chip transformation of the DNA arrays into the protein arrays. So whatever DNA you are in vitro minimalizing it, then that transforms into the protein. So this was helps you in so as I think in the lecture you have gone through that how they had developed it, and how you could understood it. So from the transcription and the translation from an immobilized DNA template. So in the PISA they were putting into sorry array, they were cascading through your particular modifications like style modifications or like this. So here what is that? So in NAPA the expression plasmids containing the proteins is GST functions are GST fusions or biotin and you're immobilized into blood cells with avidin and anti-GST which acts as a protein capture reagent. Then is used in situ expression of the proteins. So it helps you in the in situ expressions of your proteins using rapid reticulocyte lysate or your wheat germ extract, so that it gives all the necessary requirements for the translation and provides good quality with limited lateral spreading. Variation in the quality generated by the system. Okay, so 8 different cell cycle proteins from the density of 512 spots, you can understand, and ranging from different. Okay, so here also in PISA and NAPA, those both helps you to arrange your protein arrays in situ, and it removes any concerns about your protein stability during storage or the expression system and how it will be removed, purified, everything just gets out. Okay, so mixed with the different GST fusion proteins with their corresponding avidin and capture antibodies. So just uh, look at the differences in both PISA and NAPA, and how they are. So I think in the corresponding lectures you will get detail about NAPA about more, but still, if there are something questions. So one thing I'd like to stress here is that if you want to immobilize certain proteins and want to capture it or the antibodies, and if you want to detect it, then one thing is for sure is that you need to have your structures quite well preserved, otherwise it won't get interactions, won't happen quite easily. So the protein structure becomes quite important, so the immobilization techniques will try to see that will be considered as the best one which doesn't disrupt those protein structures quite well. Okay, so this is the one thing. And then your detection things, level free detection. So here you can go through that MS based detection for the disease based for the cell D of MS and cell D, and that matrix as just I have told you about different ionization techniques, and uh, about the mass analyzers. So you can go through it to understand the different time of flight ionization mass analyzers and the different ionization techniques. Okay, so if you go through it, I think you can understand the whole of the microarray, how the biomarker research is and how it is helping to understand. And then comes another important application for your is drug discovery. So it helps you identify that if you screen the small molecules, your protein array chip, so then will give your inhibitors you can identify on the basis of the interactions. Okay, so I'll just share the link with this, so if you can go through it, you will not only get a very good concept, also I will share this things also, so you can get an experience of the application of what you were studying. Yes, another thing which I think it will help you in further subsequent things also. So it will help you for that actual thing regarding NAPA, that how it has helped the addition of cell extract of the GST fusion tag, and then the mRNA protein and the capture tag with the antibody. So but and in the site will through your coated surfaces. So here you can see that this, like this, it will get the chips, and then you can just analyze all those. So NAPA alternative methods and also just okay. So these are the major thing I think you have gone through the week one, and these are the major things that you have gone through, which helps you with understanding. So I have told about the mass analyzers and the mass spectrometers, some basic things, then about the ionization techniques. Then we have gone through the different microarray applications, the different technologies, and the immobilization technologies. So immobilization techniques have been just derived from your DNA microarrays. So you need to capture and immobilize those proteins. So then the usage of these proteins becomes quite tedious from the in an expression vector system. So you need to have it from then came the cell-free expression system, and also there was affinity tandem affinity for the identification of the interactions. Then what are the applications? So there is your antibody detection thing in the microarray. Another thing what is there is that for the functional annotation or functional or regular interaction understanding microarrays. So these are the two major divisions. So once you can detect it, for diagnose it, and for screening of serum profiles or anything proteins. So these are the two major things, and also there are certain principles on your first of the which will quench and which will produce your uh, that interactions will help you in the expressing certain proteins in the promoters and of. So do you have any doubt or anything? Okay, so if you uh, don't have any doubt or anything to discuss more, you can leave. But the meet will continue till 5 p.m. After that, we will close that.
जी थैंक यू। उसका लॉक हो जाता है अंदर से। आज